Hi, my name is Sebastian Teuns, and together with Professor Hans Nauwink, we are the co-founders of PathoSense, a new spin-off from Ghent University. And in this video I will explain the problems that we see with the current diagnostics of infectious diseases and how we want to solve them with PathoSense. Now, when we look outside to the diagnostics of infectious diseases, we see actually three major problems. The first problem is that as a veterinarian, you always have to make a prior selection of which pathogen you want to test for. This means that in your head you always have to already make your diagnosis before you can select some specific tests to screen for the presence of certain pathogens. Now, if we have a limited budget of €100 to spend, we can maybe pick up 2 or 3 PCRs to perform a screening, but in reality the situation is often more complex and different pathogens can be present in this type of complexes. This means that if we select the wrong pathogens to screen for, we will end up with an inconclusive and negative result. The second problem that we see is that veterinarians are, of course, very critical. We are not only interested in a positive or a negative result, we also want to know information on the strains that we pick up with the diagnostics. Let me give you some concrete examples. If we pick up a purse in the lungs of a pig, we want to know if it's a wild-type strain or a vaccine strain. We also even want more information because we want to see if the strain has been the same that we picked up last year. If it's not, then it means that we have a biosecurity problem. And also we want to relate this information to vaccine efficacy. And this is the case for different viruses and bacteria. Now, if we look to these bacteria, then the problem is also challenging over there because picking up a bacterium, just a positive result of an E. coli, for instance, is not enough. We also need to demonstrate virulence factors to see if the germ that we isolated was really causing the disease. If we also look to antimicrobial resistance, then we are lagging behind for some pathogens. Some pathogens are really difficult to grow, and it can take up to 1 or 2 weeks to really provide information about antimicrobial resistance, for instance for Brachyspira and different Mycoplasma species. Now, it's also a long-term goal to really develop tests that can be used fast and to give in one day a complete overview of the resistance patterns of such difficult to grow organisms. And the third problem is that the standardization of sampling is actually lacking in the field. As a veterinarian, you're standing there by your patient, which can be a herd or an individual dog or cat, and then you have to decide which organ systems you have to sample. Now, this is extremely difficult sometimes. Another problem is that the type of material and how they are conditioned can be very variable. And this means that this all has an important impact on the quality of the diagnostic analysis. Now, this is our solution. So, a complete sample collection to diagnostic interpretation platform for infectious diseases in veterinary medicine. Now, the first thing that we developed was actually a new patented swab. A swab which you can use to not only collect your samples but also immediately purify viruses and bacteria inside that sample. Now, in the next step, you as a veterinarian will register the client and clinical information in an easy-to-use mobile app so that you don't have to fill in paper sheets anymore. Now, we take care of the transport of the samples so that they arrive in a fast and cooled condition at the PathoSense laboratory. Now, in this laboratory, we use third-generation sequencing to give a full overview of all the pathogens that are inside the sample. And for this, you don't have to make a prior selection anymore of which pathogens you want to test for. Now, we report all the results also in the mobile app, and we'll give you a semi-quantitative overview with a short interpretation on what you can do with this information. Now, of course, a lot of novel things and also complexes will appear on the radar. And this is also important then to guide you and to educate you with the PathoSense community. Now, how does the analysis technically work? Because this is quite important to understand, because this is not a PCR test. With a PCR, you will go to select specific primers for each specific pathogen. Now, this system is completely different. Now, using the sampling kit that we will provide, you will be able to rapidly deliver pure and really clean samples to our lab. And if you wouldn't have this system, it will lose you a lot of time to perform the analysis, because then we have to do a lot of manual steps with also a high chance of cross-contamination between samples. Now, with this sampling system, we can really deliver the pathogens rapidly to a third-generation sequencing technology, and this is shown here. So this is the Oxford Nanopore Technologies flow cell that you see, and this is actually the size of a stapler, so very small. Now, in this flow cell, we have a membrane, and in this membrane, we have around 2000 nanopores. And a nanopore is actually a protein of E. coli, a hole in this membrane. And now what will happen is that there will be an ion current flowing from one side to the other. And this is being measured by a chip in this flow cell. Now, the DNA of our pathogens will also be going through this nanopore and will cause a disruption of this ion current. And then this signal is being measured again and can be translated into a specific nucleotide sequence. And this information we can then use to perform a broad screening of the pathogens inside your sample. The system was developed first for viruses, and in autumn 2020 we also were able to add mycoplasma species. And now currently we're further improving the system to also give you the opportunity to screen for bacteria. So this allows you to give a full analysis with just one test. Because we also have a lot of genetic information about the pathogens inside the sample, we can also give you already a lot of information about the viruses that you find. Some concrete examples: if you have a purse in the lung sample of a pig, we can tell you if it's a vaccine strain or a wild type strain. And maybe if we have enough genetic information, we can even give you a lot more further genetic information. If you look to influenza, it will be possible to determine the hemagglutinin and the neuraminidase genotypes for these specific strains. And this is possible for many different viruses. Also in R&D, I think it's important to stress that it's our long-term goal to really keep on improving this test. So because we want to add a lot of new features in the future. I think very important will be to also give you information about resistance and virulence of bacteria. And this will help you forward in a very targeted treatment that can be installed very fast. Because the platform is not PCR dependent, it is of course very useful for not only one specific animal species, but it can be used for many different species. We have here the typical production animals, also the pets, the companion animals, but of course also some exotic animal species, like here some pitons or even aquaculture. So if you want to have more information, just send me an email at sebastian.teuns@pathosense.com, and I would be very glad to set up a meeting with you to discuss your interests. Thank you very much.